Several ways of detecting nucleic acids exist, and branched DNA assay is one of them. This technique amplifies the signal in a very interesting fashion. Idea is very simple. Let's suppose you want to detect some mRNA with known sequence. What would you offer to amplify the signal from a single mRNA molecule? It would be not so bad to use several small complementary oligonucleotides conjugated to some fluorescent molecule. They will multiply the signal when hybridized to this mRNA. But branched DNA technology utilizes some kind of chain of hybridizations. It has the first level of amplification called target-probe pairs. These oligonucleotides tightly hybridize to mRNA, and they have regions complementary to the second part of this complex construct called preamplifiers. They are also oligonucleotides, and they also have several regions complementary to the third part of this system called amplifiers. This chain of hybridizations ends with fourth level of oligonucleotides conjugated to the fluorescent molecules. Branched DNA complex can provide a theoretical 8,000 to 16,000-fold signal amplification of the targeted mRNA. By only changing the target probe pairs, one can amplify the signal from any mRNA. Of course, more sensitive quantitative PCR exists, but branched DNA assay applies to completely different cases. Branched DNA assay is often coupled with flow cytometry. It allows us to measure the level of some mRNA, cell surface, and intracellular proteins simultaneously in each particular cell. So one of the main advantages of this method is the detection of specific mRNA on a per-cell level. Also, it allows us to label cell targets for which antibody regions do not exist at time. In its simplest form, the combination of branched DNA technology with flow cytometry can be employed for qualitative and semi-quantitative determinations, such as the characterization of cellular targets of viral infection, with a concomitant quantification of their viral load. This technology is also ideal for correlating mRNA and protein levels, for studying their expression kinetics, their respective half-lives, and also for verifying the effectiveness of mRNA silencing or regulatory interventions. The ability to measure gene expression by flow cytometry creates many opportunities for efficient investigations of nucleic acid expression characteristics in heterogeneous populations. If you want to know more about branched DNA assay, let me know in the comments below. Thank you for watching. Good luck!